So the topic or the title of my talk is Addressing Cancer's Unmet Needs. And this is a couple words that Joanne pulled off of our website and suggested as a title. And I said, that sounds interesting. I think I can run with that. So what I'm going to talk about is our general strategy. I'll give a little snapshot of some of the data that we've generated. But in general, it talks about why we sort of think we're different from some of the other companies out there, in particular some of the other peptide development and therapeutic companies. That is me. That is some funky lines. Hopefully we'll.
Yeah, right. I did not save it to the laptop. Maybe that's the problem. It's around here.
So there should be a graphic there which shows all of the possibilities. Oh, okay, so we're not good. I can talk anyhow. Initially, I show a graphic which shows all of the possibilities for a therapeutic intervention. It shows extracellular receptors that are very well addressed by antibody therapeutics or large molecules. It shows some intracellular kinases and such that are really nicely targeted by small molecule therapeutics. But then it shows in red there are a couple boxes that are not very well addressed. And those are what we refer to as some of the unmet needs of cancer. And one of those major boxes is transcription factors. Transcription factors, of course, are important for binding to DNA and expressing particular genes that our body needs in normal physiologic scenarios.
There's something, some of the slides will not come up, but most of them.
Okay. So. However, cancer has hijacked a lot of these transcription factors to do its dirty work, to express genes that promote survival, that promote proliferation, that repress differentiation, that promote metastasis. And these are obviously hallmark characteristics of tumor cells. So what we tried to do at Sapience Therapeutics is we tried to address these unmet medical needs or unmet targets by using peptide therapeutics. We are a peptide discovery and development company. Typically, we target these intracellular protein-protein interactions that drive oncogenic transcription. We have multiple programs. For our lead program, ST101, which is currently in IND-enabling studies, targets a particular transcription factor complex of the ATF5 transcription factor and the CEBP beta transcription factor. These two, again, are normally, they are activated in physiologic conditions, but generally only under development. Once cells differentiate, they lose particularly the ATF5 transcription factor, and this is shut down. Tumor cells hijack this. We have developed a method to inhibit this process from going and therefore impact the cancer cells themselves, leading to cell death. This is a challenge. Okay, so this is the strategy of how we interact with these transcription factors. Essentially what I'm trying to show here is that transcription factors complex together. Before they bind to DNA, they do their work, which is transcription of genes, and in the case of oncogenic transcription factors, they express these proteins which promote the tumor cell to progress. What we try to do is we try to inhibit the association of the transcription factors, therefore preventing complex formation and therefore shutting down this entire process. And we have an iterative process that we use to develop the inhibitors of these transcription factor complexes. And this is a description of a library approach that we use. It is not by any means a very pretty slide at the moment. However, what it tries to show is that we have a system where we can express in bacteria cells, which is a great model system for us, whether a library of hundreds of thousands to millions of potential hits interact with a target. We can easily discern those that interact because those bacteria, the interaction itself confers survival to those bacteria under selective conditions. We therefore pick those bacteria, we grow them up, isolate them, grow them up, and we take out the DNA of those bacteria and we sequence it. And from that sequence, we can identify the peptide hits that bind with the target. We then have an iterative process. Wow. Okay. We then have an iterative process where we take those hits and we put them through what we call our knowledge-based design. And that does multiple things to the peptide hits. We add a sequence to the peptides which enable them to get into particular cellular compartments. So we have cellular penetration domains that we use to get our peptide hits into the nucleus, get our peptide hits into the cytoplasm, or wherever they may be most effective. And that's based upon what the target is, what the target does. We then, generally our peptide chemists take the molecule and they look at the potential interactions with our target. And what they try to do is they try to enhance the specific interactions while inhibiting any potential side interactions. And we do this by understanding protein-protein interactions in general. We do this by understanding peptide-protein interactions. And what this does is it gives us a small panel of potential hits that we screen by multiple different methods. We screen in vitro in cell-based assays. So generally we'll look at whether if we're inhibiting an oncogenic transcription factor that's important for cell survival, we'll look if inhibition of that transcription factor complex results in cancer cell death. We'll also look at the transcription of particular genes that are expected to be expressed by that transcription factor and if we can repress that gene transcription. This becomes an iterative process. We can take our small panels, we can then see the hits that result in positive or beneficial changes to the peptides, combine them, subtract them, add additional changes until we get our lead optimization stage. And what this slide was supposed to show is what we're able to do is take a peptide that has activity, but minimal activity is just basically a proof of concept, and move it to a therapeutic or pharmaceutical grade peptide which has enhanced activity, it has enhanced solubility, it has enhanced manufacturability, stability. We can put this into cells or ultimately into animals, get the information we need to show that we are impacting the signaling pathways we target and therefore getting the readouts such as cell death or tumor cell kill. And this is a demonstration of one of our molecules. This is our lead molecule, ST-101. ST-101 works against a panel of human cancer cell lines. Here we're looking at cell lines that range from breast cancer to glioblastoma, leukemia, lung adenocarcinomas, as well as malignant melanoma cells. And on the left, we see that exposure to ST-101 results in a low micromolar EC50 value. This is a method of determining sensitivity to a particular compound, in this case our peptide. A low micromolar EC50 indicates that at these concentrations, we're looking at 2, 3, 4 micromolar, 50% of the cells die by apoptotic cell death or a particular cell death process. And if we look at, in contrast, if we look at normal human cells, which is on the right, peripheral blood mononuclear cells, bone marrow-derived mononuclear cells, we see that these cells are not responsive to our peptide by any means in the same dose ranges as the cancer cell lines. So what this is a nice demonstration of is selective cancer cell killing by our peptides. And this fits with the paradigm of targeting oncogenic transcription factors, which promote pro-survival proteins only in those cells that have the signal. So if a normal cell has that signal, it doesn't overexpress these survival factors. The cells are not dependent upon these survival factors. So if you inhibit the system, which really isn't active, it doesn't have any impact on the normal tumor cell, on the normal cancer cells. If we take this molecule, and here's the next slide indicates that if we take this molecule in vivo into tumor models, and the tumor model that I showed here was going to be a glioblastoma brain tumor, which is a very difficult to address unmet medical need. We show that in our tumor models, we have a very significant impact on tumor growth. In fact, we have tumor regressions that persist well after exposure to our drug. And in fact, we've, in the particular study where I was going to show, had tumor cures in over 50% of the animals tested. So we've taken a molecule from a concept, we've run our library process, we've enhanced the pharmaceutical characteristics of our molecule, we have taken it into cell culture, looked at its potential activity, we've gone back and then we've modified the peptide even more based upon those in vitro activity profiles. We've come to a lead molecule, which is now in IND-enabling studies, which is, in other words, that is the set of studies that address efficacy and toxicity or tolerability of our molecule to put together a package to go to FDA and to bring this molecule into human clinical trials. And now we are well into that process. We expect to be able to file our IND about a year from now and be in clinical trials in patients with solid tumors in about, shortly after that time frame. So we've taken a molecule and we brought it this far. We hope to get to these next couple steps and come back, next year and the year after and show some nice clinical data as well.